Hi class, today's lesson is for module 1. It's the one flip lesson that you need to watch for this week. Every week I will most likely have a flip lesson for you. Some weeks I may not, depending on the activity we do. Okay, I'm gonna go and share with you guys my desktop so that way you can see see me in the video. I think it's important to stay connected during this time. Normally I don't have my face on here. And also, so you can see where I navigate to pull everything up. Okay, so you notice that on this flip lesson, I still use the topic numbers that we use for our lessons while studying for the IGC exams. I will still continue using these numbers just because I think it's good for you guys to know where I've taken them from. This lesson has been slightly adjusted and changed compared to the one that you can find in Lessons and More. Okay, so today's topic's on inheritance. We're going to run with this topic for a couple of weeks just because there's a lot of really great activities associated with it, and I just think it's super interesting. So we're going to cover inheritance, chromosomes, genes, and then how proteins are synthesized as a result of the genes that we possess. So we'll start off with a do now. Here you have a picture of a bunch of babies, and I want you to think about, I want you to answer these questions. State what makes this baby human. State what determines its gender, okay? State what determines its traits. So pause there, think about it, come back. All right, hopefully you've pretty much just said genes, 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 and that is correct. So for the, making a human baby versus a giraffe, a baby giraffe or a baby, I don't know, ant, it's the genes that we have, right? Now, all of the materials that make up all of our genes are the same materials, regardless if you're a bacterium versus a human. But the way it's sequenced, the way it's laid out will determine then what kind of genes an organism have, and then ultimately what the organism is. You can look behind here, this baby back here doesn't look very happy. Okay, in terms of our gender, it's still the genes. To be really specific though, it has to do with the chromosomes X and Y and X and X. And we'll talk about that in a little bit as well. And then our traits, going right back to our genes, right? Now again, we all have the same ingredients that makes up these genes. It just depends on the sequence that we lay these out that will result in the different genes we have. So the traits, the characteristics, the features, these are all passed on from generation to generation in the form of genes. So we inherit these from our parents. So if you look at a picture of me and my parents, you can see very clearly that my height is a result of my parents, right? Both of them are quite small. They have the genes for shortness, for height, and they've given it to me. So I want you to think about and describe all the traits that you inherited from your parents. There was this one time when in a parent-teacher conference at a school I was working at, I was meeting a parent for the first time, and there was like a bunch of parents kind of walking around, and this one was walking towards me, and I thought right away, I knew exactly whose kid this parent belongs to because they looked identical to each other. Okay, now we know then that physical appearance, as you saw my parents and me, that it can be inherited from one generation to next. But what about things such as like personality or aptitude? Have you ever heard of someone saying, man, like, yeah, I get my hot temper from my mom, or like I get my laid back, personality from my dad, or whatever it is. Now, what do you think? Do you think these kind of things are as a result of being inherited through the means of genes, or is it because of the environment you happen to live in? So same thing with aptitude, right? Aptitude is like your IQ, your ability to grasp and understand things. So it really depends. Now, it is believed that this is passed on, that it can be passed on by genes, but it can also be further shaped by the environment you're in. And you see this, you see evidence of this in twins, like identical twins who were separated at birth. So identical twins separated at birth, living in different environments. So even though they are living in different environments, they come together again as adults, for whatever means, however they found each other, and they found that the personalities are quite similar, the mannerisms are quite similar. Or you have identical twins who are separated, and due to one having different opportunities in education and nutrients, they were able to possess like a higher IQ, for example, right? Okay, so here are your learning outcomes. This is I have eliminated many learning outcomes compared to what I would normally teach for IGCSE. I really should also include and talk about haploid and diploid numbers here. So even though you don't see another learning outcome for that, I will speak to them. And I know you guys already know them because of topic 16. Okay, if you never heard of the Human Genome Project, super, super cool. So I asked a question here whether you think the human gene, like all the genes that we possess, have been fully sequenced and identified. So hopefully your answer is yes to that. And this happened a little while ago, probably around the time that you guys were actually born. So back in April 2003. And this was like a huge collaboration with many different scientists from around the world, and we were able to get a blueprint of all the genes that humans possess. And this is really important because when you think about trying to solve problems, trying to figure out how do we get humans that might be sick due to a genetic disorder, how do we make them then healthy again? So thinking about ways to like edit our genomes, edit our genes out. So literally taking genes that are in a genome and snipping them out so that way they're no longer causing those unhealthy features. Okay, so when we think of inheritance then, this is the transmission, the passing on of genetic information from generation to generation. And we're doing this because of the DNA that your parents are giving you. So remember what DNA is again, right? DNA is a biological molecule. We've learned of like 4 major ones, right? Carbohydrates, proteins, lipids, nucleic acids. DNA and RNA fall under nucleic acids. So if you remember what DNA stands for, right? It's deoxyribonucleic acid. And within our DNA, that structure, you can find then bases that are sequenced that will result in a certain gene that you have that will code for a certain protein that will result in a certain trait that you possess. Okay, looking inside of our nucleus then, inside our cell, you can actually see chromosomes or your DNA depending on what cell cycle that you happen to be in. So if your DNA is tightly wound together in this thread-like structure called a chromosome, you can actually see this. And the DNA goes in the structure when it's ready to divide, so during like cell division. Because again, you want all your DNA to be duplicated. And that's why sometimes you can see the chromosome in an X structure because the other half is just an identical copy of the first half. And in that way, when the cell goes to split, the same exact copy of the genetic material is found in one cell and found in the other cell. Okay, so again, chromosomes, it is a structural form of that DNA, and it carries that genetic information that we find in genes. So just to kind of simplify it, you look here, each of these different each of these bands represents like a different gene that's found on there. Okay. A gene then, again, it's just the length of that DNA that codes for a protein. This picture here, this image, sorry, is a photograph underneath a light microscope. And I believe it's from the root tip of an onion. So if you see here, all of these boxed off areas, one cell, we kind of saw this before when we were looking at that one lab way, way, way back at the beginning of this school year. And right here, you can see copies of chromosomes that are splitting into separate sides of the cell because eventually it's going to be separated into 2 different cells. So you can see them underneath a microscope, which is pretty cool. So ideally, eventually we want to get to the understanding of how proteins are made. So how do we go from this genetic information all the way to this protein that is made? So here's our cell, here's the nucleus. Inside, again, you have all that genetic material. And then from that information, okay, depending on the sequence of the genes the sequence of the bases that makes those different genes will result in the coding of a certain type of protein. So we know that the DNA controls function. It controls the production of proteins. For example, enzymes that are being made, antibodies that are being made. We already know what those are. Receptors that are found along cells that will accept like neurotransmitters. So it's really, really important that we have the right sequencing for the right type of proteins that we need. Okay, so there is a gene for every single protein in your body. So again, here's your DNA. Segments of length within that DNA shows all the different genes that are on there. Now, we make a little copy of that, which is known as the messenger RNA. This happens inside the nucleus. That gets passed out into the cytoplasm of a cell, where it then will code for certain amino acids that come together to make a certain type of protein. Because remember that, right? Remember that proteins are made out of the subunits, which are amino acids. So humans have roughly 20,000 to 25,000 different genes in them, but we're not expressing them all at the same time. We're not always making all these different proteins. It depends what our bodies actually need. All right. So I mentioned earlier that there are different types of proteins that are made from our genes. So for example, hemoglobin shape. Do you remember hemoglobins, those proteins that are found in red blood cells? They are controlled by a sequence of bases that make up a certain type of gene. So this could be really good for you, but also really bad for you, because if the sequence of the gene results in a clump-shaped hemoglobin, that leads to a type of red blood cell called sickle cells, which is not good for you. And if you get both copies from your parents to have sickle cell, it can lead to death, right? It usually does lead to death. But for most of us, right, especially in this part of the world, we have normal hemoglobin, so normal shaped, meaning that the sequence of the sequencing of the amino acids we have resulted in a certain type of protein that is, in this case, a normal shaped hemoglobin. Okay, so again, normal shaped hemoglobins result in our normal shaped red blood cells. Clumped hemoglobin results in something called sickle cell. You can tell that these ones then are not going to be very good at carrying oxygen. Okay, just to look at this again, if we go inside that DNA, okay, we're pulling up the DNA and we're making a copy of an mRNA from it, and then that's what eventually leads to that protein being made. So here's the thing. Remember that in DNA, you have apples and trees, AT, cars and garages, CG. But when you're looking at an RNA molecule, it's only half the strand of that DNA. And you'll also notice that it doesn't have any T, it doesn't have thymine. So instead of a T, they have something called a U instead. So that means that instead of adenine A always being paired up with T for thymine, for RNA, you will always replace the T with the U, okay, and that is for uracil. This will make a lot more sense once we start looking at how to make proteins. Okay, so I know this looks really, really scary, but let's try to simplify this. Over here is the bottom part of, or whatever, the top part, a part of the nucleus of a cell. So here's the nucleus region, here's the nuclear pores, and outside here is the cytoplasm. This hamburger structure here, the top bun and the bottom bun, that is a ribosome. Okay, remember that ribosomes, their job, their function is to make protein. So it's for protein synthesis. All right, inside here you have your DNA. Remember this long structure of DNA. And DNA has two sides of the strands. All these little color like pegs, like they look like teeth. Remember, those are your bases, right? So like ATG or ATGC. Okay, so what happens is in the middle, so some part of that DNA where there's a segment of a gene, okay, it unravels and it opens up. And by opening it up, it separates the pairing of those matching bases. So remember, A and T and C and G. So it separates it up. And what this allows is it allows for the free nucleotides that are floating inside that nucleus to come and pair up with one half of that DNA molecule. When this happens, you get a new structure that's made. So you can see it right here. Okay, there's one nucleotide, there's a free nucleotide coming in and pairing up. And what you're making here is an mRNA. So remember, being an RNA, when you have an A on the DNA strand, you can no longer pair it up with a T, but instead you replace it with a U. So even though in this diagram, you don't see the little teeth-like pegs coming out and shown here, like as you see here, they are there. And you can see it really clearly down in this image here. Okay, so step number one, all the genetic information, the coding is always found in the nucleus, right, in that DNA. Okay, that's important to know that. Step number two, an mRNA, okay, is made and it carries a copy of that particular gene, so this one right here, out into the cytoplasm. So it passes through the nuclear pore and comes out into the cytoplasm. where it meets up with a ribosome, okay? Once it gets to the ribosome, it's step number 3, okay? Remember, all the mRNA has the pegs coming off of them. It passes through the ribosome, and when it does this, as it's passing through the ribosome with each of its own base, a different type of RNA called an nRNA that carries a certain type of amino acid, depending on the 3 base it has, it will bring over the amino acid and start linking them up together to make a protein structure. Well, first, remember, it makes a polypeptide, and then depending on the properties of those amino acids, will kind of bound together, will form together to make that protein. Okay, so are we good so far? Hopefully that makes sense. Okay, so that's number 4. So again, that protein that is made is due to the specific sequence of those amino acids that come together, and that is due to the specific sequence of the mRNA bases. So for example, if you see here, the mRNA, it's AUG. AUG results in TAC because it's the opposite, well, not the opposite, the pairing complementary bases, and that will bring in this particular amino acid that will join in. Okay, and the next one gets joined in, and the next one gets joined in, and eventually that long polypeptide. And that's how you make a protein. Okay, this is just another way of looking at the image. Again, here's your DNA, it becomes unwound, the mRNA down here gets made, mRNA leaves, passes through the ribosome, that tRNA comes along, brings in a specific type of amino acid that joins up to make that long polypeptide, eventually making that protein. Okay, all right. So for the last step there, number 5, I want to be really clear on this. When you look at the that mRNA in its sequence, right? Depending on the sequence, it will result in a certain amino acid coming in and then eventually making that polypeptide. So for example, if you've got the sequence of AAA, so adenine, 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 that will go and result in a tRNA bringing in a lysine amino acid. At this point for you guys, you don't need to know the different types of amino acids. I want to put some context here so you understand that. Here's AGG, okay? That particular 3 codon of those bases result in something called arginine being made, okay, another type of amino acid. So if you see here, these are different types of amino acids that are brought over. So their actual chemical structures here, we just simplify it by drawing a sphere. Okay, don't worry about this. This is just a different ways that the 3 different codons, like a basis, can come together to make different types of amino acid. So just ignore that. All right, here again, we have a chromosomes. We recognize these different bands are our genes. Now, we like to use the word locus as the location of a gene. So, hey, here's the location, the locus of, let's say, the gene that codes for eye color. Here's the locus that, codes for a gene for hair color, and so on and so forth. Okay, remember that every individual from the same species will have the same genes, but do we all look the same? Absolutely not. And that's because there's variations within those genes, and because not all genes are expressed at the same time. Okay, and we'll talk about that variation now. So there's 2 chromosomes here. These are paired chromosomes because you get one from your mom, one from your dad. When they pair up like this, okay, we call them homologous. So homo, same, right? Homologous chromosomes. Many genes then have different alleles. I mentioned before that we have variations between us because we don't all look the same. And that's because we have different versions of the gene. So we all have a gene for, let's say, eye color, but we have different versions of the gene for eye color, and that is known as your allele. So for example, I have probably alleles for brown eyes because my eyes are brown. If you have blue eyes, you probably have alleles for blue eyes. Okay, so again, an allele then is a version of a gene, so different versions of a gene. So for example, again, a gene that controls eye color, let's say it's found here, one from mom, one from dad, but you have maybe different alleles for that eye color, maybe brown, maybe blue, okay, resulting in this different traits that you actually then physically see. from earlier, we talked about hemoglobin shape, a shape that controls the shape of hemoglobins. You can have alleles for normal or clumped hemoglobin, resulting in this actual physical trait that you see. Okay, hopefully that makes sense. All right, so just another way to look at that, okay? Here's your two genes, sorry, here's your two chromosomes, one from mom, one from dad. Maybe dad gives you the allele for brown eyes, okay? Maybe mom gives you the allele for blue eyes, okay? Depending on which one is the stronger one, which is the dominant one, that's the one you end up actually seeing in front of you. Okay, and then almost just to kind of wrap it up, I want to talk about sex chromosomes. So those are your X and Y or X and X. So the first 22 pairs of chromosomes are like your autosome chromosomes. All right, the last two, so the last pair, the last two chromosomes, the last pair are known as your sex chromosomes, also known as your allosomes. So again, if you look at this individual, right, they have an X and a Y chromosomes. Can you state whether they're male or female? Okay, hopefully you know that that is a male. If it was XX, two long ones here, then it would be a female chromosomes. Okay, sorry, female individual. The animation is a little wonky on this one. Actually, no, I think I kept everything. I just wanted to add the letters in. So we know that's an X chromosome, that's a Y chromosome, this is a female, then this is a male. So can you determine which chromosome, the X or the Y? Actually, no, I want to rephrase that. Can you state which parent is the parent that will determine, that will give you the chromosome that determines the gender that you become? So which parent ultimately determines the gender of the baby? Hopefully you know that it's the male, right? So your mother, no matter what, will always give you an X chromosome. But your father can, your mother from her egg. Your father, though, in the sperm can either give you an X or a Y. So if it happens to be a sperm that is carrying the Y chromosome, you end up having a male, a boy, right? If you end up, if the father ends up giving you a sperm with an X chromosome, you end up with a female chromosome. Okay, and then just lastly here, talking about the haploid and haploid and diploid numbers again. So a haploid nucleus is the nucleus found in gametes, those sex cells, right? Because there's only half the number of chromosomes. And then in the actual zygote or the organism themselves, that's two haploid nucleus coming together to make a diploid nucleus. And therefore, that is the one that carries both sets of chromosomes. Okay, the last little thing that's back here is just a what's called a karyotype, and a karyotype is something that we can actually, it's really cool, we can actually print this off. So you can take samples from the mother's amniotic fluid, and then from there you can actually look and take pictures of the chromosomes that are found in there and then pair them up. This is really important because then you can see whether there's any non-disjunctions that are happening. So any errors in the number of chromosomes. Sometimes you might see like an extra chromosome on number 21. So instead of having just 2, you have 3. And that results in an individual known as Down syndrome. So these are great because we can actually then take a look at what we are potentially if you're pregnant, potentially what type of, what type of baby you might end up making as a result of the number of chromosomes that are found. Okay, I was trying to find a political correct way of saying that. Sorry, guys. All right, so I'm going to leave that there, and hopefully that makes sense. Please make sure you're asking me questions on Wednesdays during help session. So sign up for the time slot that works best for you.